Hello, and welcome to this Microbiology Bio 203 video lecture from Chapter 13 of your OpenSTAX book, Control of Microbial Growth. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. Learning outcomes for Chapter 13 include understanding the control of microbial growth definitions, physical methods to control growth, and chemical methods to control growth. We begin today by examining how to prevent or stop microbial growth. One of the first things to look at are fomites. Fomites are items used by humans that can harbor microbes and aid transmission. There are a number of things that we touch and deal with in our day-to-day lives that fall into this category. Illustrated here are just a few from your car. To protect ourselves in a clinical setting, laboratory biological safety levels are maintained. This table summarizes the BSL levels from 1 through 4. In our lab environment, most of our work will be done between BSL levels 1 and 2. In hospitals and clinical labs, BSL levels 3 and 4 may be achieved. This is an example of equipment that might be used in elevated BSL level 4 environments. Types of control of microbial growth on fomites. To protect yourself from microbial growths on structures that you come in contact with on a daily basis, activities such as sterilization, disinfection, and sanitization can reduce and/or eliminate microbial loads on these structures. Sterilization involves the destruction of all microbial growth, including viruses. Disinfection destroys pathogens often on surfaces. It can eliminate vegetative pathogens and uses both physical and chemical control methods. Sanitization simply lowers microbial counts to safe levels, but does not destroy all microbial life. It is usually used in commercial establishments and involves things like high temperature washing or a quick dip in a disinfectant. Types of control of microbial growth on living tissue involves activities such as antisepsis and degerming. Antisepsis removes microbes from living tissues. Antiseptics are used for this and usually include chemicals to control the growth of microbes. Degerming is the mechanical removal, but not killing of microbes from a limited area. Swabbing the skin with an alcohol prep before injection is an example of a degerming activity. Which protocol do we use to protect ourselves from microbial growth? Simply put, it depends. It depends on the item. Critical items are used inside the body or penetrate tissues or blood. These items must be sterile. Semi-critical items contact mucous membranes or broken skin. These items need a high level of disinfection but no sterilization. Non-critical items contact but do not penetrate the skin and simply need to be cleaned. This table will provide you with a summary of the protocols, their definitions, and activities associated with them, including the agents used to achieve each of those protocols. Take a moment to familiarize yourself with each of the protocols listed in this table before moving on in the video. Sterilization. Sterilization, again, is the destruction of all microbial life, including viruses. It has a variety of medical applications and commercial applications. In commercial sterilization, objects are sterilized to the point that would actually kill the spores of Clostridium botulinum to prevent botulism. Other precautions in the medical field include sterilization of instruments, the use or utilization of aseptic technique, and the maintenance of a sterile field when working on patients. Microbial control, or stopping growth or killing microbes. Two terms should be studied when considering microbial controls. First is a biocide or germicide, and second, bacteriostatic. Biocides or germicides kill microorganisms. Fungicides kill fungus, virucides kill viruses, and bactericides kill bacteria. Bacteriostatic agents simply inhibit growth and multiplication of bacteria. The suffix stat or stasis simply means stop. If bacteriostatic agents are removed, then microbial growth may continue, as opposed to bactericidal or fungicidal activities, which would require recolonization of a surface by a microbe or fungus in order for growth to continue. This is a graph of the microbial death curve. Microbial death curve illustrates death at a constant rate in what we refer to as the D value or decimal reduction time. The decimal reduction time is the amount of time it takes to kill 90% of the population. The decimal reduction time is used to calculate exactly how long a surface might need to be exposed to a control agent in order to achieve stasis or cidal outcomes. Factors to consider when choosing a method. First, the initial size of the microbial population, the time of exposure, the environment, and lastly, the characteristics of specific microbes. Physical methods of biological control, starting with heat. Here we will look at how heat controls microbial growth. Heat denatures enzymes. There are different ways to kill with heat. We might use moist heat, pasteurization, or dry heat. Laboratory media, glassware, hospital instruments are all usually sterilized by heat. Anything that is heat labile or sensitive to heat cannot be treated in these ways. Dry heat kills by oxidation effects. Flaming, incineration, and hot air sterilization are examples of dry heat sterilization. In dry heat sterilization using hot air, a hot air oven can be used, which can heat objects up to 170 degrees Celsius for as long as 2 hours. Dry heat is effective, but takes longer than moist heat. The air doesn't transfer heat as well as water. Moist heat, or autoclaves. Moist heat produces steam under pressure, so temperature is greater than that of the boiling point. The standard for an autoclave is 15 psi, which achieves 121 degrees Celsius and can heat objects for as little as 15 minutes in small batches to achieve sterilization. There are many tabletop and industrial versions of autoclaves that are effective at sterilizing a wide variety of objects. One key is that the object must be open to the environment inside of the autoclave. Sealed objects, while they would be sterilized on the outside, would not be sterilized on the inside and might actually explode if not left open. This is an example of an autoclave. You will not be tested on the working parts of one. Pasteurization. Pasteurization eliminates pathogens and lowers microbial numbers to prolong the life of a product. Classical treatment for pasteurization includes heating a material to 63 degrees Celsius for as little as 30 minutes. There are high temperature short time pasteurization processes as well, which raise an object's temperature to 72 degrees Celsius for about 15 seconds, and ultra high temperature pasteurization, which can actually sterilize a liquid like milk. This is a good process for areas where refrigeration is not available. This is an example of the pasteurization process as used on something like milk. Refrigeration. Refrigeration reduces the temperature of a medium to slow or reduce bacterial growth. Saprophytes will continue to grow in a refrigerated environment. The bacteria may not grow as quickly as they may simply become dormant in the low temperatures. Some bacteria may actually die. Some bacteria can be preserved by freezing while others are killed. Eukaryotic microbes are often killed by freezing. This is an example of refrigeration and freezing used to preserve or extend the shelf life of a variety of products. Pressure. High pressure can kill microbes. Pascalization is the implementation of high pressure processing in the food industry. Hyperbaric oxygen therapy is also an example of pressure being used to kill microbes. This process helps infected patients. It increases tissue oxygen levels, stimulates the immune response, and helps fight oxygen-sensitive or anaerobic bacteria like Clostridia. Desiccation is a scenario where the hypertonic environment causes plasmolysis in the microbes. The microbes themselves are simply dried out. And lastly is lyophilization, where all the water is removed from the microbe, oftentimes done in situations where organisms are exposed to pressure and extremely low temperatures. This is an example of some different types of hypertonic environments as they are used to treat things like food. Ionizing radiation. Ionizing radiation comes in a variety of different forms. A couple of examples include gamma rays and X-rays. Ionizing radiation involves the use of high-energy electron beams. These electron beams ionize water and make hydroxyl radicals, or OH negative molecules. These molecules react with cellular components, particularly DNA, and cause excessive mutations which kill the microbe. There are pharmaceutical and healthcare uses. This is even used in the post office and can be used for food. Here's an example of ionizing radiation being used on fruit. Non-ionizing radiation is used in lab hoods. There are germicidal lamps that can reduce and or slow microbial growth in laboratory hoods. Ultraviolet light is often used in these cases. UV light cross-links thymine molecules and DNA. The unfortunate side effect of the utilization of UV light in lab hoods is that it is not a penetrating form of radiation. So ultraviolet light is really only good on surfaces. Filtration. HEPA filters can remove microbes, endospores, and viruses as air flows through them. Membrane filtration can do the same thing for liquids, removing bacteria from liquids. And it is good for liquids that cannot be autoclaved because they're heat sensitive. This table will provide you a summary of the physical methods of control in the previous slides that we've just covered. Take a moment to pause the video and review the different types of physical methods of control and their applications. Chemical methods of microbial growth control. Chemicals can be used to control the growth on living tissue and inanimate objects. Most chemical controls do not sterilize, but simply reduce the population or remove vegetative cells. An appropriate agent must be selected for chemical control methods to be effective. No single disinfectant is appropriate for all circumstances. The type of agents, item integrity, safety, expense, and ease of use all must be considered. First, we'll look at phenol and phenolics. These were first used in surgery by Lister decades ago. They are lipid-soluble and damage cell membranes. They can denature proteins. They are still used at approximately 1% in throat sprays. They are stable and will work in the presence of organic matter and are excellent disinfectants for surfaces. Cresols are derived from coal tar and are used in Lysol. Bisphenols, which include things like hexachlorophene, are good agents against gram-positive bacteria. And triclosan is an antibacterial found in soaps, toothpaste, cutting boards, sheets, towels, chopsticks, high chairs, and even mattresses. It's good against gram-positives and gram-negatives. Heavy metals. Heavy metals have oligodynamic action. That means in very small amounts, they can exert antimicrobial activities and denature proteins. I've listed some examples for you here, which include silver nitrate, mercury, copper sulfate, and zinc chloride, and where they might be found. Silver nitrate is used to prevent ophthalmia. Mercuric chloride prevents mildew in paint. Copper sulfate is an algicide, and zinc chloride is found in mouthwash. Halogens. Halogens include liquids such as iodine. Tincture is a solution in aqueous alcohol, and iodophore is a combination with organic molecules, like what we find in betadine. Iodine oxidizes cellular components to achieve the reduction in microbial growth that we desire. Chlorine. Chlorine is a halogen. Halogens exhibit oxidizing characteristics that shut down cellular enzyme systems. Chlorine gas, bleach, and chloramine are common forms of chlorine that are used for reduction in microbial growth. Alcohols. Alcohols, most common form used in microbial growth, include ethanol and isopropanol. 60 to 95% concentrations of these alcohols is needed to achieve bacterial growth control. The alcohol penetrates the bacterial cell wall and denatures the proteins inside. This can kill bacteria and fungi, but not endospores or non-enveloped viruses. Alcohols are good for degerming. The microbial control comes from wiping away dirt and microbes along with skin oils. They act and evaporate quickly and leave no residue. Alcohols are commonly found in things like hand sanitizer. Surfactants. Surfactants include soaps and detergents. They lower the surface tension of water and create emulsions and are effective for degerming. Quaternary ammonium salts, or quats. These are cationic detergents and they disrupt membranes. They are stable, non-toxic, inexpensive, colorless, odorless, and tasteless. They have a bactericidal effect on microbial growth, but do not affect mycobacteria or endospores. Bisquinides, like chlorhexidine, cause gross permeability changes to the cell membrane. They're used on skin and mucous membranes, combined with detergents and alcohols. They affect vegetative bacteria, yeast, and enveloped viruses. Mycobacteria, endospores, and non-enveloped viruses are not affected by them. Alkylating agents. Alkylating agents inactivate enzymes and nucleic acids. They include things such as formaldehyde and ethylene dioxide. Formaldehyde is a strong, broad-spectrum agent which can sterilize objects. It is, however, an irritant and a carcinogen. Things like ethylene dioxide are gaseous sterilizers, also very effective, but explosive and again, carcinogenic. Peroxygens. Peroxygens are oxidizing agents that are highly reactive. They oxidize cellular components of microbes. They include things such as ozone, hydrogen peroxide, benzoyl peroxide, and peracetic acid. Peroxygens can be used in water treatment. They can be used for contact lenses and food packaging. They can even be used and acne medication. Their application determines the quantity or concentration that will be used, and the desired end product will determine the volume and time of exposure. Supercritical fluids. Supercritical fluids are applied in a compressed state. Carbon dioxide at 10 times atmospheric pressure can act as a sterilant. It can enter cells and form carbonic acid, lowering the cell's pH. It's useful for food, medical devices, and tissues. Chemical preservatives, such as sulfur dioxide, sorbic, benzoic, propionic acid, sodium nitrate, and the like, are all good microbial growth controls. Sulfur dioxide is used in winemaking. Sorbic benzoic acid inhibits enzymes and reduces pH. Sodium nitrate prevents germination and growth of botulism endospores. The effectiveness of germicides. Germicides' effectiveness will range from high to low. High-level effectiveness kills vegetative cells, leading to sterilization. Intermediate level is less effective against endospores and viruses. Low level kills only vegetative cells and certain enveloped viruses and are ineffective against endospores. Again, to take into consideration when looking at what process, what chemical, time of exposure, and concentration to use, one must consider the environment, the concentration of microbes, the temperature, the time of exposure, and pH. Phenol coefficient. Phenol coefficient has been historically used and compared to phenol. Its use is centered on the testing of organisms such as Staphylococcus aureus and Salmonella. Phenol coefficient of 1 is equal to phenol. The diffusion method. The diffusion method is another tool used to determine the effectiveness of a treatment on microbial growth. Here, microbes can be spread on a plate, and a treatment solution can be placed within that on pre-soaked filtered discs. The plate will be incubated, and after a fixed time period, the researcher will determine where the lawn grows. The extent of the zone of inhibition, correlated with the ability of the disinfectant, will help you determine the effectiveness of the disinfectant that's being studied. This is an illustration of the diffusion method. Notice the white or clear areas around each yellow disc. The greater the zone of inhibition, the more effective the treatment. E-tests provide a minimum inhibitory concentration. Use dilution tests. Use dilution tests involve metal cylinders that are dipped in broth cultures of test bacteria and then dried. Contaminated cylinders are exposed to disinfectant for desired time periods. The cylinders are dropped back into the broth. Growth or no growth is recorded. This will tell us if whatever you're studying kills the bacteria or not. In-use test is used to check if disinfectants are contaminated. Here, a disinfectant is mixed with an inactivator. If growth is observed, the disinfectant was contaminated. This brings our coverage of Chapter 13, Control of Microbial Growth, to a close.